Hello and welcome back to the Pittcon podcast. In this episode we speak to Dr. Philip Bevilacqua about the world of RNA chemistry. Be sure to like, follow and subscribe to the Pittcon podcast wherever you get your podcasts.
Hi, I'm Phil Bevilacqua. I'm a professor of chemistry and biochemistry and molecular biology at Penn State. I'm also the department head there, and my research is in the general area of RNA chemistry and RNA biology.
Great, and talking about RNA biology, how has interest in, as well as our understanding of RNA, changed over the past 10 years?
Yeah, I've been myself working in the RNA field all the way back from when I was a graduate student and throughout my independent career, which has now been 25 years. And then I'd say it's been super exciting all that time. The last 10 years has been a real explosion in our understanding of the importance of RNA and biology, from microRNAs and CRISPR for gene editing, both of which have won Nobel Prizes. And then, of course, with the pandemic, the SARS-CoV-2 virus is an RNA virus, and then the vaccines were made out of RNA, made out of mRNA. So it's just been an incredible period of time for RNA.
No, definitely. And talking specifically about these different kind of forms of RNA, what is a tautomer and how does it relate to RNA's chemistry?
So there's been a huge amount of interest lately in modifications to RNA. So RNA is actually a really simple polymer. It's very similar to DNA, which almost everybody is familiar with. There's only 4 different side chains, and they look pretty similar. There's kind of 2 sizes. There's a big one and in A and G, and smaller ones in C and T for DNA, or C and U for RNA. So and they look pretty similar, so there's not a lot of sort of molecular variation there. And so there's been an explosion of interest in covalent modifications to RNA. So where RNA is modified with a methyl group or an acetyl group, and that regulates gene expression. What our particular symposium is on ways in which RNA gets modified non-covalently, and that is an example is a tautomer. And in tautomer, the smallest and the simplest element is hydrogen, and the hydrogens move around as protons or proton transfer. So they move from this position up to that position. And while that might not seem like it's that important, it's actually critical because it can change base pairing specificity and refold RNAs.
And what are the usual methods that you'd use to investigate these RNA tautomers?
Sure, yeah. So there's 2 broad approaches. One is computational approaches, where we look at databases of RNA structures that have been submitted to databases, and we analyze those on the computer or do simulations with force fields like molecular dynamics or even quantum mechanics. And then the other approach is purely experimental, is to go into the lab and try to measure differences. And those experiments, my lab is primarily experimental, although we do some of the computation, and that can be how the bases react with small reactive molecules. And if there's not a hydrogen, it might react, and if there is a hydrogen, it won't react, or it will react with a different species. That's one way, and the other are different types of spectroscopies in which we use electromagnetic radiation, and it interacts with matter, and we can get information that way too as well.
And talking a little bit more about the first method you mentioned, what are the benefits of combining these computational and wet lab experiments to investigate RNA tautomerism?
I think it's computational chemistry has come so far in the past, let's say, 25 years. Also, there was a Nobel Prize in chemistry, I think it was back about 5 years or so, for development of computational methods. And so it allows one to investigate problems that are very, very difficult to do experimentally, if not impossible to do experimentally. So we can move these hydrogens around, for example, and ask which structure is more favorable, more likely to be populated. But of course, we still need to do the experiments to see how it really comes out. And it's really the marriage between the two that is really where the strength is.
And are there any challenges that you've either faced yourself or that you've seen in the field with investigating these RNA molecules?
Yeah, there's lots of challenges. First off, RNA is not a particularly stable molecule, and so working with it in the lab is always challenging. It can degrade from enzymes that are in the cell or in cell lysates will degrade the RNA to it's chemically not very stable. So we have to take lots of precautions when working with RNA to do these types of experiments.
And why is it so beneficial to broaden our understanding of RNA chemistry, particularly in the context of understanding how biological systems have evolved over time?
Yeah, I mean, certainly there's just doing basic research, which is always really important, just curiosity-driven research. How does RNA, which is such a simple molecule with just these 4 side chains, enhance its diversity? But then this is important for many practical problems, human disease, cancer, so on, so that we can understand better how RNA might refold and that might change gene expression. And then how that can be targeted with small molecules, with therapeutics. Almost all of the conventional drugs that we have and that we use now target proteins. There's only a very few that target RNA, and those that do tend to target ribosomal RNA. And yet, the vast majority of the genome is transcribed into RNA, but is not translated into proteins. So there's this vast array of druggable molecule in RNA if we can sort of unlock how to do that. And one of our speakers, Amanda Hargrove, was focusing on that particular aspect.
No, it certainly is a rather interesting area of genetics at the moment, isn't it? And what are you working on right now that you think is like the most exciting?
Yeah, so what I talked about today is one of the areas that's particularly interesting, and that is looking at whether RNA is adopting these different tautomeric states or different charge states. So where it's not always neutral. So the bases can pick up a proton and become positively charged, or drop one and become negatively charged. And we've been studying that for almost my whole career, but what we've developed over the last decade here at Penn State with a number of colleagues, especially my colleague Sally Osman in the biology department, are ways to use next-gen sequencing technologies to investigate reactivity of RNA, not just in one transcript or one functional RNA that might only be 100 nucleotides, but looking across an entire transcriptome that might be tens or hundreds of millions of different bases. And so new technologies to basically increase our odds of finding these rare but really important states.
That sounds really interesting. So talking more about your attendance at Pittcon today. For the last 2 years, Pittcon had to place its face-to-face events on pause because of the COVID-19 pandemic. But what benefits can be kind of gained from attending these face-to-face events?
I guess I'm a bit old-fashioned, and I feel like the interactions between people are much stronger, and the connection's much stronger when they're in person. And so it's exchanging ideas, it's developing new collaborations, and ideas that I feel like only happen when people are face-to-face.
No, definitely. And what are you most looking forward to for Pittcon San Diego next year?
I haven't gotten that far yet, but probably the fact that it's in San Diego, which is a lovely thing. And I think one of the exciting things about Pittcon is always seeing all the new technologies, all the new instrumentation. I would say in my own research that it's about 50% technology development and about 50% application to biological problems. So it's always exciting to see that aspect of things.